Hello everyone, I am Professor Charu Dogra from Ramjas College, University of Delhi. I will be starting with a new series today related to the microbial world, and today we will be talking about the overview of microbiology. So, the learning objectives for today's lecture would be to get an overview of this microbiology as a discipline and to know about the history of microbiology and contributions of some renowned scientists. Now, I did give one lecture on microbiology initially, and you can find it in the channel. It was basically, we talked about a little bit about the microbes and everything, but today we will talk it as a discipline and we will start with the course series for Microbial World. The references for this particular session is Pelczar Microbiology and the Microbiology and Introduction by Tortora and Case. It's basically for educational purpose. Now, I ask you that all of you, do you recognize this? I am sure, 99.99% of you would be able to recognize this, as it put all the world on its knees. Yes, this is coronavirus. Now, this coronavirus, we have very traumatic memories of this coronavirus, which basically infected many people across the globe, irrespective of their citizenship, their caste, their creed, their religion, their sect. They basically infected anyone, and it was very, very traumatic. Now, this one coronavirus made not only the biology people, but even the non-biology background people to know about many of these terms, such as hand wash, of course, became the routine thing, and it was always a routine thing, but we've got focused on this hand wash a lot. The social distancing, infection, quarantine, sanitizer, COVID-19, pandemic, vaccines. So, the virus, when we say this, we generally think that all the study about this is restricted to the people who have taken biology as a discipline. But this coronavirus and COVID-19 made the entire world, irrespective of the educated, non-educated, literate, non-literate, biology background, non-biology background, everybody to know about these basic terms such as virus. Even people got to know about the technique of RT-PCR, which is a biotechnological technique, when there was used to detect the coronavirus in the infected individuals, when they used to take the swab from the nasal cavity and then they used to do the PCR. So, people came to know about that. So, what I want to emphasize here is that you have an idea that microbes are the organisms which are not visible to the naked eye. So, the organisms which we cannot even see, and one organism, this virus, put the entire world on its knees. It came to a standstill. There was so many, non going out and everything that happened. So, you can imagine the mighty power of this unseen organisms. Now, just to get to tell you about certain numbers, just the weight of one coronavirus is approximately 8.5 into 10 raised to the power of minus 21 kg, which is per gram, which is 8.5 attograms. So, that is the weight of one coronavirus. Now, number of viruses needed to make a person sick. I am just telling you certain numbers to appreciate one fact and to understand about this mighty power of these unseen organisms. So, around 70 billion viruses are needed to make a person sick, and therefore, the weight of these 70 billion would be that much gram per gram. The worldwide cases of COVID-19 infection at one point of time were around 2 billion, that 2 million, that's what we have taken, just one example I am taking. It later on became much more, but for example, at one particular time, the coronavirus infections, the COVID-19 infections were basically 2 million people. Now, the weight of total number of viruses infecting this 2 million people, if you calculate, it comes out to be less than 1 gram. So, I really want you to appreciate the fact, or at least recognize the fact, that less than 1 gram of coronavirus made this entire planet, stand still. The entire planet came to on its knees with just a gram of virus taking over. So, the organisms which we cannot even see from the naked eye, we cannot just ignore them. They are very mighty, they are very powerful, and as we will see in our subsequent lectures, that they actually rule the planet. In fact, in the human body, we do not have as many number of cells as many we have microbes living on and in our body. So, you can imagine the strength and the functioning of these microbes, which are very, very essential to understand. So, microbes are basically the living organisms, that is microscopic, the textbook definition, and it can be visualized only with special instruments such as under a microscope. So, you can see certain microbes have been shown here, the bacteria, then there is these even amoeba. You can see the pseudopod, which is trying to engulf a food particle. Then the second one is basically the mold. So, you can see the spores which when fallen, that is the most common observation you must have had, that when you have your bread or your chapati lying for a number of days, it develops, there are certain greenish kind of a thing that grows onto it. And these are basically the bread molds, and these bread molds, when you look under the microscope, these are high resolution microscopes, they appear as filamentous, they appear as hyphae. So, these are again microorganisms. If you just observe pond water under microscopes, you will see the Volvox, which is on the top right hand corner, and then underneath it is the Zika virus, which is infecting a nerve cell. So, you can see these are the variety of microorganisms which are there. We will talk a little more about that. We did talk about the bacteria in our previous one of the lectures that I started with earlier, but we will talk about each of these in detail. But what I want you to know in this slide is that these are the organisms which can only be seen under a microscope, and these are all microscopic images that you can see. So, this microbe term was first suggested by Maximilien Paul-Émile Littré, the great French lexicographer, the one who makes dictionaries, and linguist who was also trained in medicine, and proposed to the Académie Impériale de Médecine by the French surgeon Charles Sedelot, and quickly adopted by the French school. So, just for the information, that is how the microbe term came into being. Now, when we talk about microbes, what we basically observe is that these microbes, our perception is that microbes is basically the germs, the kitanu, what we talk about. They are the one that makes us sick, they are the one that spoil our food, they are just the bacteria and viruses that we know. We get cold, we get flu, we get all the infections. So, what we have this perception is that these microbes are all the villains, just like the Bollywood movies, right? So, COVID-19 pandemic even strengthened this view that the microbes are the villains, because as I said, less than 1 gram of a virus made the entire world come to a standstill. So, we have that perception that these microbes are bad. But let me tell you this, that we have to change our perception. We have to change our perception because the harmful microorganisms are way less than what beneficial microorganisms that we have. Again, we will discuss a lot about these in our subsequent lectures, but there are so many advantages and beneficial role that these microbes play, and without which also, the entire planet will come to a standstill, or it will just not work. All the ecosystems will disrupt if these microbes are not there. So, many benefits are there, and therefore, we have to change our perception that these microbes are not just the enemies, but they are the superheroes. They are doing a lot of good work as well. Now, coming to the microbiology, as the name suggests, microbiology is the study of microbes. So, study of living organisms that are microscopic, and they include bacteria, fungi, algae, viruses and protozoa. and encompasses their beneficial uses and detrimental effects in disease causation as well. So, not just knowing about what types of bacteria, fungi, algae, viruses are there, and even the protozoans are there that are microscopic, but also to know about the benefits, the uses, or the detrimental effects in disease causation. So, this encompasses the entire microbiology. Now, looking at the microbiology disciplines, we have generally, we do not divide it as it is shown like a flow chart here, but I just want to emphasize that you know about these certain terms, that microbiology deal with the living organisms which are basically microscopic and cannot be seen with the naked eye, as well as there are certain agents which are infectious agents which are not living organism per se, but they again are microscopic and cause the various kind of diseases and also are beneficial to some extent. They have certain roles, some of the organisms. So, these are basically dealt under these various disciplines. So, microbiology is a discipline under biology, and these are then disciplines when we talk about and deal with living organisms microscopic, such as bacteriology. We deal with phycology, mycology, which is the study of algae, phycology, mycology is the study of fungi, the parasitology, where we basically talk about the harmful organisms which are the parasites, or protozoology, where we talk about the microscopic protozoans. So, these are the various sub-disciplines of the microbiology discipline. And then we also talk about the infectious agents, where the major one is the viruses, and therefore there is virology, where we study about these viruses. And then we also have certain infectious agents, we will talk about them, such as viroids and prions, which are usually harmful, and they are also studied under the microbiology discipline. As I said, we just do not talk about the types of these organisms, or basically they are just habitat or niche, but we also talk about the processes they govern. So, even in the macrobiological processes, where these microbiological organisms or the agents are playing a role, that all comes under the microbiology discipline. So, this is the overview we are talking about the microbiology. So, the microbiology encompasses all these things. We talk about the food microbiology, where we study about the beneficial microbes which results in the various kinds of foods that we take. You know, one of the example again which is very primary is the curd that is made by the lactobacillus. So, we all know that the milk gets converted into curd by the addition of the lactobacillus which is there, and other fermented foods which are there, the batter that you make for idli, dosas, even all these things, the fermented food, as well as we talk about the foods, the microorganisms which are harmful for the foods or which spoil the foods, like bread molds. So, we understand how these mechanisms are governed, how we can modify them, and more knowledge about them. So, that comes under like food biology, microbiology. Then there is again environmental microbiology, where we talk about the microbes which are present in the environment. They are present in the soil. Again, there are beneficial microorganisms which we will talk about in the detail, such as the the primary grade information that you already have is the microorganisms that live in the root nodules and of the leguminous plants and fix the atmospheric nitrogen and increasing the fertility of the soil. So, rhizobacter. So, those organisms we talk in such examples, and we also talk about the organisms which are basically making the environment non-friendly, non-habitable. So, some of the pathogens can also be spread through the environment, the soil, the air, the water, the contaminated water. So, all these infections can also be spread through that. So, we talk about the environmental microbiology. There is agricultural microbiology, where we particularly talk about the the organisms which are pests, or the organisms which are beneficial for the growth of the plants. then there is industrial microbiology. Then coming to the processes, these are the applications disciplines of the microbiology. If we talk about the processes, then there is basically the enzyme microbiology, or there is genetics and genomics and everything. When we talk about the how what is the makeup of these microorganisms, as well as what are the processes that it governs. So these are interdisciplinary in nature as well. When we talk about molecular biology, then molecular biology is talking about the molecules, which is the biomolecules, the DNA, the nucleic acids, the carbohydrates, the lipids and the proteins. So molecular biology as a discipline is not just restricted to microbiology. But when we talk about microbes, then it is molecular microbiology. But when we talk about, for example, in case of cell, then it becomes a cell and molecular biology. So, but we also under microbiology, when we study these organisms, and some of them are unicellular organisms also. So, when we study the different biomolecules under them, or when we talk about genetic manipulations, or when we talk about genes, so genetics, genomics, molecular biology, all of them also come under the discipline of microbiology, where the organisms that we are studying are basically the microbes. So entire repertoire of these different disciplines, sub-disciplines come under the microbiology. Immunology is a major thing, enzymology is a major thing. So how microbes cause a particular disease, how does the immune cells basically work together to stop the infection, how these immune systems can be modulated. So all these things also come under the discipline of microbiology. So microbiology is a course where the central organism is microbes, but we talk about the types, that they are bacteria, viruses, and then further their subtypes, and then their habitat, their niches, their processes, as well as their applications. So that comes under the microbiology as a discipline. Now let's look into the brief history of microbiology. Now what happened in 1665, after observing a thin slice of, we know about this, we have talked about this in our cell biology lectures as well. As I said, that the first observation, even the simplest microbes are unicellular organisms, and they are very minute, just like cells. You cannot see them with the naked eyes. So the first observation when the cell was observed was by the scientist Robert Hooke. Everybody knows that, right? So Robert Hooke reported that life's smallest structural unit were these little boxes that he observed in the cork cells. Now they were dead cells. They were not living cells. Later on, he observed, and others also observed them in the living, but they were dead cells when he observed a thin slice of cork under whatever that crude microscopes or the lenses arrangement that he had, and he could observe these cells, and that marked the beginning of the cell theory. Now cell theory, again just to remind you, there were Schleiden, Schwann, Virchow, and they were the ones who basically framed the cell theory, that the cell is the basic structural and functional unit of life or any living organism. And then later on came that the postulate that one of them was all the organisms are, all the living things are made up of cells. The cell is the basic structural and functional unit of life, and then the third tenet came in, which tells that the cells exist from the pre-existing cells. But to start with, the first tenet was that all the living things are made of these cells, and that came from the observation of these cells by Robert Hooke by these crude microscopes in a thin slice of cork. So there basically where it started from. And this is just to show you that it was basically published in this end. You can observe that that is how it looks like, just like that honeycomb kind of a structure, and cell basically means a compartment, and therefore Robert Hooke basically called them as cells, these smaller repeated units, and they said that these are the structural unit of this particular anything that is. So initially it was basically the cork or the cambium that he took. Then came Dutch merchant and amateur scientist who was Anton van Leeuwenhoek, who was probably the first to observe the microorganisms through the magnifying lenses of more than 400 microscopes he constructed. So it is very interesting that Leeuwenhoek is the one who is called the father of microbiology, but he was a very amateur scientist. He was a Dutch merchant actually. He used to deal with all these lenses, and with those lenses he constructed these different arrangement of lenses to form the crude microscopes. And when he observed just that the water, he saw certain organisms which were moving. He saw certain things. I wouldn't say what organisms. He just observed that there were certain things that were moving in the water which was there. This is Anton van Leeuwenhoek, when he is observing under, and in the middle panel you can see that is a very crude microscope that he had. So he did have what is the replica of kind of today's microscopes, but he had that, there's a stage, and then on it the sample was placed, and then there were a combination of lenses, and on the right hand most side you can see the diagrams that he drew. So, the A, B, E, F, G, these were the different kind of moving organisms, and C to D is the trail of one of the moving organism, and he called them as animalcules. So, these animalcules were the first living organisms that were observed, and these were basically the microorganisms because they could not be seen in the naked eye. He just placed a drop of water there, and all he could observe was there were these creatures which were moving inside that he could observe with his crude microscopes which was there. So, these were the animalcules that he talked about. Now, the merchants and the people got interested in the fact that where did they come from. Obviously, it was a very, very interesting finding that everything they later on observed many different samples under the microscopes, and people got more and more curious about the fact that where were these small creatures which you cannot even see with the naked eye, they come from. And initial theories of this spontaneous generation was basically coming from the non-living matter. There used to be these kind of myths that toads, snakes and mice could be born of the moist soil, because they were present in the moist soil. So, they just thought that soil is giving rise to these organisms. The flies could emerge from the manure, or the maggots, which we now know are the larvae of the flies, but they could or arise from the decaying corpses, because that is where the observation was. Here I would just like to emphasize on one interesting point here, which is not just related to microbiology, but related to science. This is what science is. And when we study any discipline of science, we should always be very careful to understand about the process of science. And the process of science starts with two things. One is the curiosity, and the second is the observation. That is the scientific process where it goes out from. You look around yourself. You get curious about things. You observe certain things. When they observed that on the decaying corpses, there were certain things which were moving. When they observed that there were a lot of flies which were there on the manure, the dung, the cow dung, there were a lot of flies there. In the moist soils or in the rainy seasons, they could observe the toads and the snakes. So, the first thing is the observation, and the second thing is the curiosity. And then comes the data collection, and then comes the identification of the techniques basically, that what to adopt to know more about that. But that's where the science basically starts from. So, this spontaneous generation was a very, very important fact, that people got curious that whatever organisms we are not seeing but they are present, where are they coming from? And the debate over the spontaneous generation continued. That we will continue in our next session. Thank you.